Well, good afternoon, everyone. Welcome to our latest New York City Builds Bio Plus webinar. And once again, my name is Mitch Simpler, and my day job is serving as a mechanical partner and as the managing partner emeritus at Jarrah Spelman Bowles Consulting Engineer. Today's webinar will focus on New York City's continued investment in new life science projects, and namely the specific fact that the New York City Economic Development Company has invested an additional $38 million into new biotech centers. Once again, at New York City Bio Plus, our mission today and every day is to promote and shine the light on the life science real estate industry in New York. Our hope is that you will feel as strongly as we do that the life science industry in New York is poised for rapid growth, but constrained by a lack of available affordable laboratory space. Once again, I would like to personally take this time to thank all of our sponsors, our virtual exhibitors, and our media partners, all of whom will be spotlighted later in this morning's presentation, and without whom we simply would not be able to have this webinar. I'd also like to personally thank all of our panelists, moderators, and presenters for their time and for sharing their valuable insights in this incredible and exploding industry. And finally, a huge thank you for all of you, our members and guests, for taking your valuable time and energy for hearing our story. It is now my honor and my privilege to once again introduce Ms. Nancy Kelly.
Thank you, Mitch, and hello, everyone. I'm so excited to welcome all of you to NYC Builds Bio's first webinar in 2021, entitled New Life Science Investment Continues. New York City invests $38 million in new biotech centers. My name is Nancy Kelly, and I serve on the steering committee and as a founding member of NYC Builds Bio. And I would like also to acknowledge and thank the New York City EDC and the New York institutions who join us this afternoon: Albert Einstein Medical College of Medicine, Columbia University, New York Stem Cell Foundation, and Rockefeller University, all of whom are building a solid foundation for the New York City Life Science Cluster. And of course, the founding and corporate members of NYC Builds Bio, who make all of our programming possible. Since the announcement by the New York City Economic Development Corporation of its $500 million initiative to propel New York City's life science sector into the top ranks of life science centers across the country, everyone has been watching as new investments were made in New York City's workforce and lab infrastructure, and waiting to see what would come next. This morning, or this afternoon rather, we're pleased to host the NYC EDC and the 4 New York City institutions who will tell you about the most recent investments designed to advance translational research efforts and grow the biotech ecosystem in New York City. So welcome to all of you, and thanks for joining us today. This webinar is part of an ongoing series of online events hosted by NYC Builds Bio Plus. You can view the series on the events page on our website, and we hope you will register for future monthly webinars to be announced, as well as our new monthly member spotlight and networking series, which will start next Thursday, March 4th at 1 o'clock, spotlighting Hunter Roberts Construction Group. We are also planning monthly virtual tours of the developments happening in subclusters throughout New York City, commencing in March with Deerfield Management's 345 Park Avenue South, called The Cure. A date will be announced for this event shortly, and we hope you will join us in all of this rich programming. Now I want to introduce Carlo Yuvianco, Vice President of Life Sciences at the New York City Economic Development Corporation, who will introduce his colleague, Elsie Yao, and our panelists this afternoon. During Carlo's 5+ year tenure with NYC EDC, he has been part of the team that developed the LifeSci NYC 10-year initiative and currently remains instrumental to the plan's implementation. His past and current portfolio of life sciences programs includes city capital projects, like the ones highlighted today, as well as investments into early stage companies, workforce development programs, land use initiatives, and regional economic development. He also serves as a strategic advisor on real estate and financial incentive transactions under LifeSci NYC. In the last year during the pandemic, he was a core contributor to many of NYC EDC's COVID response efforts, including the public-private partnership with local life sciences company Opentrons to establish the Pandemic Response Lab, as well as the public-private partnership between NYC EDC and New Lab in the Brooklyn Navy Yard to design and build ventilators. Dr. Yuvianco is a chemical and biomedical engineer by training, marked by 10 plus years of academic and industrial experience prior to the NYC EDC. He is the author of numerous publications in the field of protein science and the holder of several biotechnology patents. But most importantly, Dr. Yuvianco is a born and raised citizen of New York City. Thank you, Carlo. Before we begin, I just wanted to cover a few administrative details. There's a program in PowerPoint highlighting the hosts and speakers for this meeting, and these will be posted on the NYC Builds Bio website after the meeting. The program is being recorded and will be distributed to all registrants after the meeting. If you are having technical difficulties accessing the Zoom room or other issues, please email info@nycbuildsbio.org or send a notice through the chat button at the bottom of your screen. And since most of you are participating in listen-only mode, please feel free to submit your questions via the Q&A button at the bottom of your screen. We will open up for discussion and Q&A at the end of the program and will address as many audience questions as possible. At the end of the meeting, we will also have a chance for smaller randomized groups to meet in separate rooms with our speakers, where you can open your videos and microphones to talk to each other. You can stay as long as you would like to get to know each other and share stories. So now I'll open it up to Carlo.
Great, thank you, Nancy, and thank you for the kind introduction. Hopefully that's all you'll have to hear about me and maybe the EDC the rest of the time. Today is all about our 4 illustrious awards that we recently made with City Capital under LifeSci NYC. So that's enough about me. I'd like to introduce my colleague at the EDC, Elsie Yao, who is an AVP with our team at Life Sciences and also an early colleague on the City Capital program, which includes these projects as well as the other initiatives that pertain to real estate and City Capital. So Elsie, thank you again for joining us, and I'll hand it to you in a minute. But before I do, wanted to take this opportunity before we get into it to describe to you the run of show. Today, we're going to have a journey of the development of a drug from its earliest idea and conception, and how do you prosecute that as a piece of matter, to all the way through manufacturing of that therapeutic. And these 4 projects, we feel at the EDC and are very proud for them representing sort of segments of that development process all the way through conception to manufacturing at the pilot scale. So that's how we're going to divide up the day, starting off with Columbia University and Brent Stockwell, chemical biology professor in Colleges of Art and Sciences, with a very exciting project on that will validate therapies at their earliest stage of conception, like I said. And then we will transition to a bit of a later stage development of the drug, of a drug or a therapy, with Tim O'Connor representing the Tri-Institutional Translational Center for Therapeutics, which is being operated and stood up by the Rockefeller University. And then that will transition to 2 exciting fields of development in the biologic space: stem cell therapies, and more broadly speaking, cell therapies, with the New York Stem Cell Foundation and Einstein Montefiore's exciting project. And so with that, very briefly, Brent, I'm going to start it off with you. Can you, for our audience, introduce your organization and also yourself and your role within the organization in this project?
Great. Yeah, it's great to be here, everyone. Thanks, Carlo, for that, teeing that up. I'm a professor of biological sciences and chemistry at Columbia, and I also am involved with the various institutes like Data Science Institute and Cancer Center and the new Irving Institute of Cancer Dynamics, which is more of a quantitative approach to understanding cancer, and several other things. But here I'm heading up this effort to establish the Therapeutic Validation Center in partnership with NYC EDC and some other partners that we're working on establishing. So I'll be helping to set this up, and then eventually we'll have an executive director, and I'll continue to serve on the governance council, chairing that over, as we move forward.
Wonderful. Thank you very much. And great to have you again, Brent. Tim, sort of one of the forefathers of this push for building out the industry in life sciences, together with some of the other representatives of the Tri-I. Can you introduce yourself and also your institution and the role in the project?
Sure, Carlo. As usual, calling me a forefather, you've started by making me feel old again. My name's Tim O'Connor. I've been at Rockefeller for 8 years now. I'm the executive vice president here. And my background is actually in biology, having been run my own lab as well as participated in both basic and clinical research over the years. But I've been supporting science instead of doing it for some years now, including my entire time at Rockefeller. So Rockefeller is a small biological research institution. So we are a university, but we only have graduate students in the life sciences. We don't have an English department. We don't have a football team. It's not a university in that sense. But we do train the next generation of scientists, as well as focus on our mission, which has been a consistent singular mission since our founding in 1901, and that is science for the benefit of humanity. I like to think we punch above our weight. We're a small institution with about 70 labs, but in our history, we've won 26 Nobel Prizes, including 6 in the last 20 years. So it's just an outstanding faculty here. Doing great science, and it's my privilege to be able to be here and support it. I'll just end by noting our geography on the Upper East Side, because it's important to the project, is that at 68th and York Avenue, we actually literally share a street corner with Memorial Sloan Kettering and Weill Cornell Medicine, which will lead into later my description of why this is a tri-institutional effort that we're endeavoring. Thanks, Carlo.
Fantastic. Thanks, Tim. Rick, can you introduce yourself, the New York Stem Cell Foundation, and also your role in the foundation? I think that you bring a particularly unique perspective given your background, and maybe you can touch a little bit upon that.
Sure. Thanks, Carlo. And thanks for having me today, and especially thanks for the award. It means a lot to us. So I'm Rick Monzimo. I'm Senior VP Scientific Operations at the New York Stem Cell Foundation Research Institute. I've been with NYSA for about 4 years. Before that, I spent over 20 years in pharma industry and preclinical drug discovery, initially with Schering-Plough and then with Merck, and then was able to transition out and join NYSA, which is, I guess, the small kid on the block here, but trying to hold down the West Side. Our labs and offices are located on West 54th Street between 11th and 12th Avenues. And we're a unique foundation in that, we were established in 2005, so I've been here for 15 years, surviving as a nonprofit. And we're unique in that we support external researchers as well as have our own internal research efforts and laboratories on the West Side. With about 70 researchers on the ground and about a little over 100 employees overall. And we do a mixture, again, of external support and a lot of communications with events, both for scientific community. We have an annual conference that we hold normally at Rockefeller, though going virtual this year and last year. But as well, a lot of events for the lay community to educate them about diseases and stem cells and et cetera. So. Yeah, I'll leave it.
Great. Thanks, Rick. And last but not least, Harris, Dr. Goldstein, can you share with us your role as you wear numerous hats within the Einstein community, but particularly your role in this project and also Einstein Montefiore as an institution.
Sure. I'm Harris Goldstein. I'm for this role Associate Dean for Scientific Resources. In addition, I still run a laboratory. My major interest is in immunology and particularly HIV. And I also, we have a NIH-funded Center of AIDS Research that we actually includes Rockefeller as well as CUNY that I direct. And Albert Einstein College of Medicine is been the medical school in the Bronx and well known for its research. We've had past year $220 million of NIH funding. We have over 300 investigators working in a broad array of different areas, and some of that expertise we brought to bear for this particular enterprise, Embark. And in addition, we are part of Montefiore Medicine, which includes Montefiore Medical Center and the affiliated hospitals present in the Bronx and throughout Westchester. And as a result, we really have a very diverse participation, both in terms of basic clinical and translational research that we're able to provide. And again, we provide medical care for a large fraction of the Bronx population, as well as now increasing population in the Westchester population.
Fantastic. Thank you so much, Harris. Great to have you today. So I'm going to kick things off to Elsie Yao, who again is an assistant vice president with our team at the EDC, and she's going to briefly just have some opening remarks that will set the stage for these projects and provide a little bit more context to the inception of these projects and how the EDC got involved. And so, Elsie, with that, would you mind taking us away?
Yeah, absolutely. Thank you, Carlo. And also thank you, Nancy, for having us and the rest of our panelists for joining us and sort of talking through some of the amazing work that we've grown together in partnership over the past several months to year plus. So it's been actually really exciting between Carlo, myself, and the rest of the team this year to be able to share with the broader community these 4 new investments that we've made in the biotech centers, what we at the EDC have branded applied R&D facilities. So maybe to take a bit of a step back, I know Nancy talked a little bit about what the life sciences team at EDC does at the beginning. Thank you so much for the kind words. I also wanted to sort of give some who are maybe in the audience who might be newer or less familiar with our work in the life sciences, a little bit of the lay of the land there. So Carlo and I are part of the New York City Economic Development Corporation's life sciences group. The rest of the group does a lot of other things in the real estate realm. Our team specifically, since 2016, has been executing on the city's $500 million investment, what we've called LifeSci NYC, to grow and advance the biotech sector here in New York. So that $500 million is really allocated across 3 investment areas. The first being in real estate, the second being in talent, and the third being really to connect academia to industry with the ultimate goal of accelerating the commercialization of the scientific discoveries we make here in New York. So over the past few years, we've made a lot of headway in the first 2 categories that we're very proud of. As some of you in the audience may know, especially the real estate front, we've activated more than 600,000 new square feet of commercial biotech space through projects like InnoLabs in Long Island City, Deerfield's Cure building in Nomad in Manhattan, and then also the BioLabs Incubator space in Soho. And then in the talent and workforce end of things, we've actually engaged an external partner, Upper West Strategies, who's been leading over the past few years the LifeSci NYC summer internship program, a paid program that has placed more than 300 students in internships, keeping some of our local talent close to home, helping them stay within local life sciences companies and ultimately contribute to the growth of this ecosystem here. So going back to the 4 new centers that we're going to talk about today, those all really fall in the last category that I mentioned of connecting research to industry. It's our very first action in this area, 38 million out of the initial 150 million of city capital that we allocate to this category. And we as the city really hope it won't be our last one. So thinking back to what Carlo and I and the rest of the team were looking at back in 2016, we looked around and we sort of saw top of mind that it was going to be very important to support our world-class local institutions, prioritize ways to help them get their science from bench to bedside even faster than they were already doing. So New York City's institutions have almost historically always been second to Boston when it came to NIH grant funding that went to our institutions. And so when we looked at that data point, we saw there was no reason why that couldn't translate into equivalent success right here in the commercial industry. So as a result, the 4 institutions we selected this year are going to be part of our founding network of applied R&D facilities, New York Stem Cell Foundation, Columbia University, Montefiore Einstein, Rockefeller University. All of them are exceptional examples of the world-class institutions we're very thrilled to partner with as we work together to accelerate their translational medicine efforts. So each of them will add unique value to our growing biotech ecosystem, much of which we'll hear about a little bit later, and ultimately help more life sciences companies incubate, grow, and hopefully stay over the long term here in New York City. So with that, I'm going to turn it back to Carlo and the rest of our esteemed panelists to speak more about each of the centers in greater detail.
Fantastic. Thanks so much again, Elsie. So with that, I'm going to transition to focus on you, Brent, and the Columbia University Therapeutic Validation Center. And I know that you prepared a few brief slides for us to sort of help describe to the audience in very approachable terms what this center will be doing and what its physical nature will be. And with that, maybe you can take it away, and then we'll get into one or two questions as a follow-up.
Perfect. That sounds great.
Fantastic.
Oh, great. There are the slides. Okay. Yeah, so when this original request came out of NYC EDC, we asked ourselves, what is that rate limiting bottleneck step in order to launch a startup company when you have a promising project, particularly in academia? And what we realize is that a lot of times, a lot of investors ask for that is hard data to get is the validation of a therapeutic candidate, a therapeutic mechanism in vivo in an animal model. And then ultimately, obviously, you want to translate that into a patient. But seeing that in an animal model, all the details of that mechanism, cellular mechanism, molecular mechanism, is something that's often lacking. And when you have that, it can be validating and convincing to investors. So our view is that the Therapeutic Validation Center will provide that final piece in what we call the preclinical validation puzzle. So we could maybe state that succinctly as shown on the slide here, the scientific mission being to define the cellular and molecular mechanism by which experimental therapeutics exert their effects in vivo, validating and defining those mechanisms. So this is at the front end of the pipeline Carlo described, but it's actually at the last stage of the early discovery process. So if you think about discovering a new drug candidate, you might have a hypothesis, or maybe you did a high throughput screen, set up a cellular assay, and then you come up with a candidate compound, you optimize that with some medicinal chemistry, you do some what's called ADME pharmacokinetic PK work, where you study how it behaves in an animal. How is it distributed? How is the compound metabolized or biologic or what have you? Maybe some preliminary safety studies. And then when everything looks promising, you want to do that efficacy validation. Is this actually effective, and does it work through the mechanism we think it should work through in the relevant animal model? And so that's that last piece that we think we can add the value at that stage, because many projects there's many ways to get to that point, but getting over that hurdle is quite challenging. So why don't we go to the next slide? And just to show you the technologies we're using here are imaging mass spectrometry methods that allow you to really look in detail at molecular mechanism in vivo. And just to illustrate conceptually how this works, imagine that I'm going to show you two different paintings. And if we just look at the bulk distribution of pixels, which is the normal way that people look at mechanism in vivo, you take out a tumor, you extract compound from it, or proteins or RNA or lipids, and then you just see how much of different stuff is there, irrespective of which cells they come from. Then you might see something like this. The top painting has a lot of yellow and not as much gray. The bottom one, the pixel distributions are shifted around, and it's hard to know really what that tells you. If you go to the next step forward, you could see that these actually represent, you can go ahead and step forward in the slide.
Carolina, yeah.
Yeah, these actually represent two very different pictures, right? One is a child's drawing, one's the Mona Lisa. And you only get that information by seeing the location of the pixels, where they are in two-dimensional space. So if you go to the next slide, I can show you this in a little more scientific detail, but that's the big picture, that location matters in terms of where cells are and where drugs and metabolites and lipids and proteins are. What cells are they actually present in? So why don't you go to the next slide? So, oh, sorry, back one. Yeah, so with new emerging methods in mass spectrometry imaging, we can actually look in detail at the single cell level now and see where a drug goes and how it's metabolized and what effects it exerts. So on the left, you're seeing an image of HeLa cells in culture, and the green dots are actually a drug that was treated, these cells were treated with amiodarone, this small molecule, and then we can see where it is inside each cell. The red is the nuclei, seeing a piece of DNA that identifies the nuclei, and the blue gives you a lipid that's present throughout the cell. So you can see these individual cells and where the drug is in those cells. And on the right is another example where you're seeing individual cells in culture. Green is showing you again the extent of the cell, the cytoplasm, and in pink you're seeing individual mitochondria by a lipid that's present in mitochondria. So we can zoom in at the single cell level and see what's there and how is it affected by a drug. So why don't you go to the next slide now? So at a tissue organ level, we're planning to use this by taking, let's say, liver. If you're treating with a drug that accumulates in the liver, maybe it's a liver cancer model, you want to see what's happening. So we can make sections from the liver and then subject the sections to different imaging modalities. We can use this mass spectrometry imaging to see where the drug is, which cells is it accumulating in, and what's changing in terms of metabolism and lipids, drug metabolites. And then in some cases, we can also look at proteins and RNA in individual cells and then put all that data together, do 3D reconstructions, and really get a molecular map of what that drug is doing in the liver or the liver cancer model. So I can go to the next slide. Yeah, so this is sort of, if you think about it from a practical standpoint, what can you do with this kind of technology? Well, you could identify drug sensitive and drug resistant populations in the relevant animal model of the disease. You can identify what we call pharmacodynamic or PD markers, meaning how do cells respond? What goes up and down in those cells when they're treated with a drug, which are things you could later monitor clinically to know if patients have received an adequate dose. We can see the immune effect of drugs. Are immune cells recruited? What kind of immune cells? How are they affected? And basically determine how the drug is accumulating in different cell types. Maybe it's all going in macrophages and that's not what you want. You know, you have to re-engineer the drug. And again, from a big picture perspective, this would give you information, allow you to go back if necessary to the drawing board, redesign your therapy if needed, and ultimately try to orchestrate a concerted response of all the different cell types for maximal therapeutic impact. Why don't we, I think that might be the last slide. Was there one more actually? Oh yeah, and so this is just programmatically, where does this fit in the New York City landscape? We view the Therapeutic Validation Center as sitting between the New York academic institutions, some life science companies, maybe small startups that want to do this type of work for their next round of fundraising, and investors who are looking at launching new projects. They can all come in with projects. There'll be an application process to enter the pipeline. And then coming out of the center, we'll be partnering these projects with investors to launch new New York City life science companies. Some of them will go into the startup incubators already around town, and then connecting them with investors more generally for their next financing. So in a nutshell, that's what we're trying to do scientifically, and we're really excited that this could add something valuable to the landscape here in New York.
Great. So thank you so much, Brent, for that explanation and a glimpse of the science that is part of the drug development process. I think that's very illuminating to a lot of the audience members today. I was wondering if you can touch on one additional aspect to the broader picture of this center that you're standing up, which is the community of academics that oftentimes, and certainly at the EDC, we've pointed to as sort of the substrate by which the foundation of life sciences and the future of life sciences in New York City exists, which is all of our academic medical centers and the scientists that reside and the principal investigators, the faculty that reside within it. Could you speak to, very briefly, the culture of entrepreneurship, of technology transfer among faculty, not just in Columbia, but across the city as a faculty member yourself who's also started companies? Is it something that we've only scratched the surface of? Is there more to do there?
It's a great question. I think there's tremendous interest in the conversations I've had individually with people. And I think this is something I'll learn a lot more about as we go around and talk to all the institutions and find out, the many people that I don't know personally yet, but I hope to get to know, what are their thoughts and how can this fit into their plans? What's the entrepreneurship passion there? But I would say that in general, I've seen a lot of interest in commercialization and launching startups, but just not the expertise. And the problem is, even just not knowing who to go ask or what's the first step. Like, you have a project, and how do you start it down that path of getting an investor excited and launching a company? And so our hope is that people will come to the center, we'll do this validation work with them, and then we'll also help them with that entrepreneurship piece of what does it take to get this out the door into a startup.
No, that would be a fantastic outcome indeed. Okay, great. Well, thank you for that, Brent. Moving on, for the sake of time, Tim, I'd like to transition to the Tri-Eye Translational Center for Therapeutics. And this project is an interesting one. It's a patchwork somewhat of a strategic anchor operations. Can you provide us and the audience a little bit more detail on that?
Sure. Thanks, Carlo. And thanks, Brent. You actually teed up ours pretty well by describing the pipeline of drug development, which is basically what we're trying to support at multiple places in the pipeline. So broadly speaking, and with apologies to experts in the audience, at a conceptual level, one can think about the drug development pipeline consisting of starting with basic research that's most often done successfully at academic institutions, transitioning into preclinical development, and then into the clinical phases. And you've all heard about, phase 1, phase 2, phase 3, et cetera. And what we're focused on is how do we take the outstanding basic science at these 3 institutions that I described to you before, and help advance that, because frankly, pharmaceutical companies are not interested in reaching as far back in the pipeline as they once were. And I think we can do it as successfully or more successfully based on the expertise you find in these centers of excellence at academic medical centers in particular. And so what we did starting in 2013 was band together with Sloan Kettering, Weill Cornell Medicine, and Rockefeller. We created an independent 501c3, not-for-profit, that partnered with Takeda Pharmaceuticals. So we had these 3 institutions along with Takeda. So we had pharmaceutical industry expertise in order to take basic research and move it along this pipeline to the point where you could still partner with the academic innovators, but advance it very efficiently in the pipeline while working up to and including animal models. At that point, a project, so TDI is one entity. It exists and has graduated dozens of projects, moved them along very, very successfully to licenses with biotech companies. That initiative caught the attention of biotech industry and pharma to the point where 2 years later, we started to fill the gap of what comes next. And that's squarely in the area of true preclinical development, where you're optimizing your compound. And we took the same entity, the 3 academic institutions, to Takeda Pharmaceuticals, and partnered with 2 venture capital firms, Deerfield and Bay City Capital, and formed a for-profit institution that basically becomes a licensee of graduates from TDI. So that's been around for 5 years or so, and also has advanced things to the clinic, to where you can hand it off to a different entity, or you can create a new co along the way or upon clinical advancement, et cetera. So what we want to do next, so those 2 entities exist, but they exist in various patchwork locations on our 3 campuses. They're not all cohabitating space. and it would be much more efficient if they did. Finally, what we don't have on the Upper East Side is a biotech incubator. So unlike tech, where you can incubate small companies in Starbucks or your parents' basement or wherever for these things, you need lab space. And even if you're a small company, you still need lab space and you need it to operate effectively, safely, et cetera. So it's a very high overhead endeavor. Unlike tech, biotech is very high overhead in terms of just the support that you need to do these things. And when you're a starting fledgling company, what you really need is just a small amount of really affordable lab space. And here's the kicker. Ideally, it's really right next to or on the areas of innovation, because what you want to leverage at that very beginning stage is access to the scientific founders and the scientific expertise. And so that's the third part of the patchwork Carlo mentioned, is a new incubator on the Upper East Side. One doesn't exist. And that hasn't stopped our institutions from developing these things over the past 8 or 9 years. Annually, these 3 institutions spend $750 million in R&D money. We've spun out over 60 companies that have raised over $3.5 billion in the last 8 or 9 years, but none of it's been on the Upper East Side because we don't have incubators. So this is a critical final part to the Tri-Institutional Translational Center. Thanks, Carlo.
Oh, thank you. That's fantastic, Tim. And I think that this model, and sometimes we forget as New Yorkers, but this model of having an on-campus incubator to service a community, a concentrated community, be it from one institution or multiple, is something that has proven to be successful for other campuses as well, right? We're seeing obviously the establishment of launch labs on the Columbia Washington Heights campus, which will be exciting. But even before that, predating that, setting the foundation for a lot of what we've been doing at the EDC is the SUNY Downstate Incubator and what it has meant to the SUNY Downstate community, and also by extension, the BioBat community. So I think this is an exciting development for you guys. And from what has happened in the city before, I think it'll be very successful. But I might be biased there, of course.
Well, we appreciate your help, and you second the point that I made, which is having a number of these in strategically located clusters is a lot better than trying to do one big one somewhere, especially in New York City, where it can be hard to get around.
Quite right. And so there's one other thing I wanted to touch upon with you, Tim, and this harkens back to you being part of those original change makers, like together with Mark Tessier-Levine, who is the former president of Rockefeller, and also members of the Bloomberg administration, is this intensifying culture of entrepreneurship on the Upper East Side. Can you speak to that at all, and give folks in the audience a sense of what is happening among not just the 3 institutions, but also the Blood Center and HSS?
Yeah, for sure. So you're back to calling me a forefather. Thanks, Carlo.
I said change maker this time around. I was a little bit.
which both of those terms are actually way too much credit. And to be fair to a lot of people, there's been efforts in this area for going back over 20 years in the city, trying to tap into the scientific expertise that's long existed in the city and figure out how to apply it locally. Mark was clearly a leader in that, as was Craig Thompson, Laurie Glimpsher. There were leaders, there were faculty on the ground who've been doing this at multiple institutions, including at the institutions where my colleagues are representing today. And it is a cultural shift, but it's something that more and more scientists are interested in doing. And part of that is actually because the science has made it possible. And Rick Lifton, our president, likes to talk about this, how 20 or 30 years ago, people working in basic science, it was really hard to take basic findings and apply them to human treatments and therapies and cures. It's becoming much more accessible to do that. For example, just a well-known example is the sequencing of the human genome. We have now findings in basic research that can go. We've got one of our Nobel laureates has gone from his model organism, which is fruit flies, into clinical trials directly, without having to go through years and other animal models. And so there's just so much more understood about that. So it's exciting, and people see their neighbors doing it and get excited. It's also generational. You see younger and younger people who see their colleagues succeeding in this. So yeah, it really is exciting, and it is definitely part of part of the culture. I think Brent mentioned getting their faculty more involved in this. Many institutions, including I think all the ones represented here, have funding for translational research that has a mechanism for helping basic scientists understand what it would take to apply their research, because the majority do. Not all of them want to be directly involved, but every one of our faculty members, we've asked them all, do you want to take this and try and apply it in translational? Some of them don't want to be involved personally, but they would all love to see it happen.
That sounds like a pretty great culture to be a part of, and it seems like it's only getting more intense. So the good things to come for sure.
With plenty of help from you guys from the EDC. So that's been a great partnership and we appreciate it.
No, we're proud to do it. And I think that with all these projects and also future projects, what we like to do is get involved in these inflection points, both from a culture of innovation, but also what it might mean for the neighborhoods that you all are anchors in. So thank you for being a partner with us. I'm going to transition now to Rick at the New York Stem Cell Foundation. New York Stem Cell Foundation, and specifically its Research Institute, is a fantastic operation that, if folks in the audience are not aware of, should visit their website and all the materials and press that is associated with it, because it is fantastic, fantastic facility, as well as fantastic work that they do there. And Rick, I'm going to hand it off to you in a second, but from what I understand, there are two sides to the service shop that is the Research Institute, and the way that it engages with both the academic community as well as, and more importantly, to the EDC, the commercial and industrial community. I was wondering if you can speak to those two sides to the Institute's operations a little bit more, and how this expansion with our funding is going to help with that.
Yeah, absolutely. Thanks, Carlos. Yeah, so one of the things we do is really to partner with people, researchers, to build human-based tools based on induced pluripotent stem cells. So in addition to our internal research, we have a lot of collaborations where we develop either disease models or generate cell lines from patient populations that can be used for better understanding the disease or direct therapeutics discovery. We're also now putting together the various pieces that we built internally to begin to do our own discovery efforts. And largely the award from the EDC is going to help us expand on that and really bring in some specialized technologies to accelerate that. But at the same time, we, as being involved in the stem cell field, realize that one of the really fantastic potentials of stem cells is using them directly as therapeutics, either as cell replacements or as therapies themselves. So, over the last couple of years, we've built a capability to generate clinical grade induced pluripotent stem cells and create, in our first project, replacement cells for blindness, for age-related macular degeneration. And so we've built in a GMP clean suite to do small-scale manufacturing for basically phase 1 trials. So we're trying to kind of come at major diseases of our times using stem cells through a couple of different lenses, and helping other industries to do that as well.
Great, no, that's fantastic, and this is really a symbol of EDC and the city's commitment to doubling down on applied stem cell research. We saw this through the Bloomberg administration. We're seeing it now through the Blasio's administration, and our continued support of the New York Stem Cell Foundation. But for those who are not as familiar with the industry itself, Rick, could you comment on where the industry is going and really the commercial applications and innovations in stem cell therapy? When will we see potentially the next stem cell company or a stem cell company really have drugs or I should say therapies and treatments in market?
Well, trials are getting started all the time. I mean, it's really exploded, I'd say, over the last 5 years or so. There are companies getting formed left, right, and center. And, depending on what application you're looking at, either trials are underway or they're well underway in terms of getting into phase 2 or phase 3. And a good example locally is Blue Rock Therapeutics, which is bringing an iPS-based therapy for Parkinson's disease into phase 1 clinical trials. They just received the approval from the FDA to do that in conjunction with Mount Sinai. And so I think, those kind of precedents will really give a lot more confidence to people who want to fund these kind of approaches. But, the whole field is starting to explode. And I think, the more attempts there are, the more successes we'll see. So in the next few years, I think we'll start to see the results of that coming out, and should be very positive.
Wonderful. And, if I could just borrow an additional 2 minutes of your time to maybe comment on the New York Stem Cell Foundation's and its operations and all of that it does, including the Research Institute, as sort of an attractant to New York City, right? And we've seen this already with your anchoring of the Hudson Research Center on 619 West 54th Street, and companies trying to be attracted to that site. But I'm curious, to what extent will this expanded operation and the Research Institute become a flame to the moth of stem cell companies to New York City, in your opinion?
Yeah, I think as our CEO, Susan Solomon, likes to say, NYCIF is a convener. So we bring together a wide, a very diverse set of stakeholders, both academic scientists, biotech entrepreneurs, and other investor types. And we try and spread the information and the data and make connections. And so I think by having kind of a central and independent facility, we're able to do that quite effectively. And so we interact with all the major universities. We have collaborations with pretty much every institution in the city, and we can bring in and connect them with other other collaborators we may have across the country. And so I think, we're in kind of a very unique position in that way of being a catalyst in many ways to these things.
No, that's great. That's good to hear, is that convening activity that is so foundational to your operation is something that I think is somewhat of the secret sauce of the foundation. So thank you for speaking about that.
Yeah.
Similarly, as we transition down our drug development pipeline, very excited to introduce our next speaker, Harris Goldstein, who is leading up the efforts for the Einstein Montefiore Biotechnology Accelerated Research Center. So, top marks to Harris and the team at Einstein Montefiore for the best branding, I think, of their particular center. And also, we at the EDC are very excited that this is one of the 4 in the portfolio that is in an outer borough, and probably the first of our investments in the Bronx when it comes to life sciences. So immensely excited to see what happens out of this project and what it will mean for Morris Park and the Einstein Montefiore community. And also, before I hand it off to you, Harris, and Carolina, maybe you can just cue up his slides while I finish off these remarks. But I'd be remiss if I didn't give due credit to not just the life sciences team, but also our real estate team at the EDC, who was very instrumental in making this particular project happen and be realized in such good quality and shape. So Warren Haggis and the team and the real estate team, along with Elsie herself, were very instrumental with this. So with that, Harris, can you take us away? Harris, I think you're on mute. Sorry about that.
I want to thank Carlo. I want to thank you and Elsie and Doug Theed and the entire EDC team for support through this process. And I also want to congratulate my colleagues on their awards. And again, I think what's great is the opportunity to work together, these 4 groups, and find areas of commonalities and synergies, because I think that's really what's going to make us even more competitive. And that's what's so important. I look forward to, again, following up on that. So our facility, we named the Einstein Montefiore Biotechnology Accelerated Research Center, which we call EMBARC. And next slide. And the mission of EMBARK is to accelerate the development of innovative CAR T-cell based and antibody biologics based treatments. And one thing that all of us have experienced this past year during the COVID pandemic is the need to rapidly pivot and develop treatments for infectious agents, which we had never even heard of before. And again, the ability of the biotechnology industry to do that really is miraculous and is only going to happen more in the future. And we see EMBARK as positioning us in New York to support companies that are going to be involved in future endeavors, as well as treatments of cancers that do not yet have curative therapies. Next. So the leadership of EMBARK, our dean, Dr. Gordon Tomaselli, is the leader of this process. Next, myself. And in addition, the third crucial part of this team is Steve Almo is the chair of the Department of Biochemistry, who provides impressive expertise in protein structural biology. And next, and Brian Pulaski, who has really been crucial in terms of putting together this project, providing financial as well as other support and creativity. Next. So next. So what's our vision? So our vision is really to develop a new model to support life science companies. And all of you are familiar with contract research organizations, where you basically want to make a protein, you want to make a drug, you want to make something, you want to do an experiment in an animal model, you go to a CRO and they do it for you. However, what we think is a major advance is now combining the capacity of a CRO to be responsive to the needs of a company with access to world-class scientific expertise that can now tell you how to make things even better. And what EMBARK will be able to provide companies with is access to Einstein researchers and the shared facilities we have at Einstein to really tremendously enhance the abilities of this contract research organization. In addition, it provides companies with access to the Montefiore integrated health delivery care system, because as other speakers have spoken about, after you do your preclinical, you need to go clinical, you need to do clinical trials, and that's what we can provide. So this integrated capacity really provides a new model for accelerating the advance of life science company products from the test tube into patients and potentially therapies. Next. And Carlo discussed as well that we are in the Bronx, and one of the limitations the Bronx has been access to the rest of the city. And in the next years, the Bronx will be having a Metro North station on Morris Park Avenue, right across the street from Einstein, that will be able to provide access to Manhattan Penn Station 20 minutes, as well as access to the entire New Haven, Boston, Washington corridor. And it is an underdeveloped area of the Bronx. Rent in the Bronx is a little bit lower than the Upper East Side, a little bit lower than the Upper West Side. And we think that's going to be a major advantage for biotech companies that don't have a lot of money to spend in order to establish themselves here in the Bronx, as well as having the association of Einstein, as well as the association nearby of EMBARK. Next. What services will we provide? Next. We want to optimize CAR T and antibody biologics binding. Well, what does that mean? Next. Well, this is a cartoon of a CAR T cell, and on your left, you see a CAR T cell, and coming out of its the cell, you see this kind of Y-shaped molecule, and that Y-shaped molecule is an antibody. That antibody is specifically able to recognize something. In this case, it's recognizing something expressed on the tumor cell that tells the body this is a tumor cell. Now, if you could specifically recognize a molecule expressed on a tumor cell that is not expressed on a normal cell, then you can potentially kill it. And the whole key to CAR T cells is identifying this recognition molecule, because if it recognizes the tumor molecule at a very high efficiency, it binds it very, very tightly, and does not recognize any cell proteins, it can specifically kill cancer cells and not kill our own cells. However, if this molecule doesn't bind the cancer molecule very tightly, or it's by accident binds to our own cells, then it could potentially have off-target effects. So the key is designing these molecules that have the optimal binding to the cancer specific protein, for example, with no binding to any other normal cellular proteins. And that is exactly the area that we will be working in terms of enhancing this process. There are many biotech companies that are developing CAR T cells, developing these recognition molecules, but we will support them in terms of making them even better. Next. And so the CAR T cells, you could make better binding molecules. These CAR T cells are highly mobile. They have potent killing capacity. If they're well designed, they don't attack self, and they work in everybody. It's an off-the-shelf process. I could take cells from somebody, convert them into CAR T cells, and give them back to the patient. And there are actually currently now 2 FDA-approved drugs, CAR T cells, that are used effectively to cure cancers that were previously incurable. And next, this is just showing that there are both solid tumors as well as liquid cancers, leukemias, that also are potential therapeutic targets for CAR T cells. It's a major area of biotech development. Next. And what services will we provide in addition? Next. So we'll be optimizing these binding molecules. Next. And once you have optimized them, you need to make the CAR T cells. You need to take a few cells that you convert into CAR T cells and expand them into billions of cells. And that is what we will be doing. We'll be doing it in GMP capacity, so you can actually now take these cells and give them directly to patients to see if they work. Next. And in addition, we'll also be making GMP immunomodulatory biologics and antibodies. Again, you're now all familiar with the use of antibodies to treat COVID. And again, these antibodies can be used that are being developed to treat many other cancers and many other infectious diseases. But again, making it in a way we can actually give it to patients to test them. Next. And in order to evaluate whether they work, we're going to be supporting companies by also having a mouse vivarium where we could do preclinical studies, testing these products in a mouse model, as well as novel humanized mouse models we've developed where the mice are populated with human immune cells and tissues that can be used as a target that predict how well they actually work in patients. Next. And finally, we also will be providing them with state-of-the-art other supports, flow cytometry and analytical imaging. Next slide. Next. And just basically our vision is biotech and pharma companies that have nascent products that need to be optimized. Next. Can come to us. We'll be accessing the expertise of Einstein scientists to enable them to optimize their products, either CAR T cells, immunotherapeutics, design optimization, and production, as well as doing preclinical evaluation. Next. And other companies or ones that we've helped now may want to move it into clinical trials. So for CAR T cells, we'll have the ability to take the vectors they've developed and then now actually go to patients, get their cells, convert them into CAR T cells, and now working with Montefiore, be able to now to do clinical trials to evaluate the efficacy of these CAR T cells against curing different types of cancers or infectious diseases. And finally, next, and this is just a cartoon of what the money is going for. This is going to be renovating 22,000 square feet and making them into a GMP CAR T cell and biologics optimization production facility. Again, there is nothing in this at this scale currently present in New York City, which we think will make it into a magnet for biotech companies. In addition, we'll also be developing this vivarium that will enable us to give companies their own private rooms that they could house mice for them, and specifically for their own experiments, and again, make it very, very available to them. So again, we have tremendous hopes that this would be a major center that will enable and support biotech companies to make products that actually will be able to treat diseases and benefit both all New Yorkers as well as people in the United States and the world. Thank you very much, Carlo and Elsie.
Thank you so much, Harris. That one quick follow-up question that I'm sure our audience members might be interested in is, to what extent is this only the first among many in a series of investments on the campus, or maybe more generally the Morris Park area, to activate life sciences and attract industry to Morris Park? Would you say that this is only the beginning, or is this yet to be seen where this will take you?
No, we think absolutely this is only the beginning. Again, our dean has a vision for developing a biotech area around the Einstein campus. In addition, many of you may be familiar with Simone, which is a real estate company that actually has a lease on a significant amount of space at the Hudson Metro Center, which is across the street from Einstein. And they actually have plans to develop 2 buildings that will be devoted to life sciences with 100,000 square feet of lab space for incubator, biotech companies, et cetera. And again, perhaps even other development. And again, what's great about the Bronx is we have space and it's relatively cheap, and we have the know-how to do it. And we really see Embark as being the anchor for that by really being what's going to be drawing biotech companies, not just to space, but the ability to have the expertise to move their products forward and test their products and get their products approved.
Oh, great. That's fantastic. So exciting, more exciting things to come from that neighborhood. Thank you again, Harris.
Thank you.
All right, great. We're just about at time. I'd like to throw it back to Elsie just to provide some summarizing remarks and take us home and wrap us up here on behalf of the EDC. But I want to just, for my part, thank you all again for listening and your interest in these very exciting projects that we at the EDC have helped activate. And also thank you to the organizers, including Nancy and Mitch and Carolina for helping today. But Elsie, maybe you can share some of the thoughts that we came up with.
Yeah, absolutely. I'm going to keep it short so we can all get to the networking part and really building those relationships that we talk a lot about. So I guess that my 2 takeaways from this conversation, and definitely from our perspective, is that these projects represent complementary facilities and services sort of along the pipeline. Tim, you talked a lot about growing the geography on the Upper East Side, Harris about Montefiore and the Moores Park area. It's very important to the city to sort of invest in these individual nodes of the life sciences across the city and the 5 boroughs, but ultimately also over the long term, we want to see that impact sort of resonate across the entirety of the city, extend beyond just one campus, one institution, one neighborhood, and for really all New Yorkers to really feel this and see the great work that our institutions and the future companies in the pipeline are doing. And the second point I sort of heard from this conversation was really sort of the theme of collaboration. Brent, I loved your last slide in your deck. I really think it spoke to everything that we're trying to get at. Reiki talked about Mount Sinai and Blue Rock and that collaboration, some of the ones that are happening at Sinai as well. So personally in the ecosystem, I've seen so much growth, so much collaboration in the past few years. We think that these new applied R&D centers will certainly be an extra step that layers on top of that, hopefully building on that community, making it even stronger. And certainly Carlo and I and the rest of our team at the city look forward to seeing where this is going to go. So I hope, Nancy, that was short and sweet. Thank you so much for having us, and I'll kick it back to you.
Yeah, so wonderful presentations, everyone, and congratulations. I mean, these initiatives are just transformative for the city, and it's so exciting to see them taking hold here. And, you know, I think each of you described how this is going to advance this industry in the city in a big, big way. So really exciting times. And as we, prepare to drop into the networking sessions, it would be really great to talk a little bit about maybe the real estate component. I know, Harris, you just touched on that, which is really interesting. 22,000 square feet of space plus a vivarium. It might be interesting for the people that are listening in to hear about the teams you have that are building those spaces out, the space for the other projects, the space you're reserving. Tim, is this on the Rockefeller campus? How much space are you thinking about? That would be great to know, and I think it would help to inform our audience here. So thank you, everyone. And to our audience, please register for our next member spotlight and networking event next Thursday at 1:00 featuring Hunter Roberts Construction. And we'll be following that with a virtual tour of 345 Park Avenue South, led by Deerfield Management, and featuring, the banking and development partnerships that they put together to actually create that facility. So be on the lookout for the date to be announced in the next few weeks on that. And we're excited to kick off our programming for 2021, and lots of exciting things to come. So thanks, everyone.